are going to focus on human biobanks today, but what we talk about is actually applicable to all biobanks. Biobanks have evolved even since we've been involved with biobanking, which has only been about 4 or 5 years. We've seen a really big difference, but nobody really knows how many biobanks there are in the world. And biobanks, when I'm talking about biobanks, I'm talking about cancer biobanks, tissue biobanks, disease biobanks, and the list goes on. It's really only limited by their application. And I read a recent article which said there are more than 200,000 biobanks in Europe alone. But the author of the study then went on to dispute that some of these 200,000 were actually collections rather than biobanks. And this is one of Dan's bandwagons. Yeah. So the diagnostic leftovers, which are stored in the unmonitored fridge at the back of the lab, that's what a lot of us think of biobank actually is. And no one's too sure what the provenance of these samples are, how these samples were collected, how they were processed, how long they've been there for, any of their associated data. However, this is the source of samples that have been used extensively in research, and this questionable quality contributes to the billions of dollars of irreproducible, and you try saying that word a couple of times, irreproducible research results. There was a study done in 2015 in the US where they said that the cost of irreproducible results was $28 billion in 2015 per year. So this is where biobanking really comes to its fore. So biobanks have evolved from the leftover collections, and they've become much more sophisticated organisations. So that's sort of the cost of biobanking. This is actually one of the more modern biobanks, and there are actually a couple of these in Australia. They hold millions of samples. Robotics comes in. As you can imagine, the computer program that goes with these samples is actually pretty extensive. So biobanks have evolved from leftover collections and become much more sophisticated, and it's through the introduction of standardisation of validated protocols for collection, processing and storage of samples that modern biobanks can maximise their quality and utility to ensure reproducibility and comparability of research results. The global biobanking market is estimated to be greater than $50 billion already, and is actually supposed to grow exponentially over the next couple of years. And the introduction of AI has added to that growth. So today we are lucky to have two great speakers with us that are very much at the forefront of biobanking in Australia. Professor Catchpoole here is incredibly passionate about biobanking, and if you want to know anything about biobanking, ask Dan, and translational research. And he's here today to share some of that passion with you. And Helen, as she was introduced, is the Biorepository Manager at Biobanking Victoria, who were the first lab that we actually accredited. So I'll hand over to Dan first.
Everyone in this room is now tasked that during the evening, move forward, they have to go up to Jill and ask her to say the word irreproducibility. All right, thank you very much for having me, and I've got to be quick. 5 minutes, I've been told. All right, thank you very much. As you can see, I run a biobank at the Children's Hospital at Westmead as part of the Children's Cancer Research Unit. I've been doing that for 25 years. And for my sins, I also have half of my time now working over at the School of Computer Sciences at the University of Technology, where I'm looking at how we can introduce computing technology, computing science, data-driven methods to understanding biospecimen data. That's why that's there. I'm going to talk about 4 things today. The importance of biobanking. What is a biobank? Therefore, what isn't a biobank? And what we are standardising and why accreditation matters for biobanking. So hold on to your seats, here we go. All right, the importance of biobanking. Notice I've started with the verb, not with the noun. What is biobanking? What are we trying to do? When you talk about biobanks, everyone starts to worry about the freezer. Everyone starts to worry about things stuffed in freezers. But I want to put to you today that biobanking is far more than just what's in a freezer, as Jill said, the freezer down the end of the corridor. All right, so what is biobanking? Well, according to the ISO standard that's come out, and there's an ISO standard that's been out for about 4 years, 5 years now, 20387, and it's quoted as saying that biobanking is the international standard. It says that it's the process of acquisitioning, get that to me at 9 o'clock tonight, the process of acquisitioning and storing together with some of the, or all of the activities related to collection, preparation, preservation, testing, analysing and distributing defined biological material, or biospecimens as we like to call them, as well as related information and data. So it's about data. So it's far more than stuffing a few samples in a fridge. All right? If biobanking was all about accrediting a fridge, we'd all have our local freezers with our chops and our peas and our veggies all being able to be credited. It's far more than that. It's a dynamic thing. So what is biobanking? It's all about actually making connections in that dynamic world and sharing information. And very briefly, this is from the health world, but you have similar sorts of descriptions could be used to describe biobanking in other fields such as agriculture, plants, environmental bio and all these sorts of microbiology. It's about making connections between the patients, in our case, who have the samples, who provide the samples, the clinicians who want to interpret them and understand how those samples are going to tell the patient what they've got, and the researchers who do the deep dives into that information that is held within those samples to interpret and to pull out that information and help us understand what's going on biologically. Biospecimens are shared between clinicians and researchers, and knowledge and consenting is done by the biobanker, connecting all three of those different people groups in that particular connection. It's just an example to show that biobanking is actually not just about a simple process, it's actually about quite a dynamic process, building up an ecosystem of relationships that exist between people. And having accreditation to actually define those relationships really helps. So biobanking is also an informatics problem. It's an informatics problem. The main reason why a donor in the health world provides you a sample, right, a biospecimen, is because they want to know what the information that's in those samples. Every one of you have experienced the stick up the nose. During COVID, we've all had, who hasn't had a COVID test? Everyone knows what it's like. That is a biospecimen. That bit of snot going off, and it's gone off. You do that because you want to know the information that's in that biospecimen. Have I got COVID? Have I not? If I've got COVID, what strain or brand of COVID is it? Is my vaccine working? That sort of thing. All right? You want to find out that information. And the only reason why scientists want those biospecimens is they want to dive deep into all that molecular information that exists in those biospecimens to understand them, to help explain what's going on, and if it's a disease, how do we understand that particular disease, right? To help them ask and answer questions. So biospecimens, what's all the fuss about? They contain information. To contain information about the collection process, the experimental extractions, the underlying genetics, the tissue biology, the treatment regimens, the individual patient, and ultimately about our disease. In other words, they explain everything about us as individuals. All right? But what is a biobank, and therefore what isn't it? These are some of the quotes that I've heard over the years, just a couple of them, which actually highlight what people think a biobank is. One was, but we're not a biobank, we're a cohort. That was from a particular person who was part of a biobanking committee, a biobanking group, but then when the government said they're going to pull all the funding for biobanks, they said, but we're not a biobank, we're a cohort. You know? So, exactly. All it is a big fridge. Up in Queensland, they were having a big announcement, a big opening of a biobank, and they got the minister in to open this biobank, and she wanted to go off and see the biobank, and she was heard saying as she walked out the building, all it is a big fridge. And the other one was quite amazing from a director of our institutions, an institution here in Sydney, was saying, why do we spend all our time putting samples on ice? And in some ways they're right, in some ways they're wrong. It is a dynamic process, but it's actually really, really important that biobanking is not seen as just putting samples on ice. It is a dynamic process. All right. So what is a biobank? This is a question that was asked, and I was able to contribute to this study of international experts. It was a commentary more than anything, it wasn't really a study. And I pointed out there that I actually hate the word biobanks because they actually mean too many things. Right? But right at the bottom there, I said, a biobank can be anything. We're so confused, we don't quite know what a biobank is. But biobanking, all right, I can't read my slides here. In conclusion, the biobanks are not special, but biobanking is. It's the act about the noun. That's right. Thank you. Anyway, I'll read it off here. But the verb, the passage of biospecimens from donor to researcher scientist is fundamental and always has been to meaningful translational research. To understand human disease, you have to study the disease. And so this is actually really, really important. The actual transition, the dynamic relationship of the samples going from a donor to a scientist to actually study them and understand them. Absolutely vital to our health. Same with specimens going from animals to help understand the nature of the diseases they have, or their breeding, or how we can make plants grow better flowers or better crops. All helps us. The transition of biospecimens from people who have them to the people who need them, who can study them, helps us understand things, brings about new knowledge. And this might sound a little bit familiar, and we have actually a common, a bit of a forgotten principle that we sometimes miss. And one is that if you go to a pathology department and think about pathology, all of us are given blood samples to pathologists. There's routine sample collection. They do scientific testing. They ask patients' consent. They have patient confidentiality. They're responsible for storage. They're responsible for quality assurance. They're using established tests with known sort of range of results, and they affect individuals. Our understanding of each individual patient, the knowledge that gained from that. Biobanks do essentially exactly the same thing. We're worrying about sample collection. It's routine. We have scientific testing. We have worried a lot about consenting issues. We have patient confidentiality that we have to be concerned about. We store samples. We have a process of quality assurance, but we're investigating new things. We're actually understanding new research and understanding new hypotheses and studying things. And if we learn from that, we will discover many things. However, the things that we do in a biobank, the research world, we do want eventually to move back into pathology. Everything that's gone on in a pathology world has happened on the back of some researcher, probably a PhD student being paid very, very little amounts of money to actually develop new ideas that have been brought into pathology. Research and pathology and research and clinical care is vital. And biobanks bridge that gap between the rigour of diagnostic testing and the freedom and flexibility and creativity of research. All right. So it highlights the issue that tissue management is fundamental activity of health. Lots of different biobanks out there. There's embedded biobanks, population biobanks, targeted biobanks, virtual biobanks, distributed biobanks, conservation biobanks, museum biobanks, archival biobanks, and living biobanks. And they're not always big facilities right down the bottom there. Some people have just got samples in a box. But that can also be often considered as a biobank. So what is a biobank? According to the standard, it's a legal entity or party of a legal entity that performs biobanking. Legal entity. It actually sounds really serious. All right? The biobank can no longer be considered just the freezer down the end of the corridor. More importantly, it needs to be seen as vital and worth investing in. Facilities and departments. It's accountable and responsible for what it's doing. It's a legal thing, and has the experts and specialists like biobankers around the place. Right? It needs to be taken seriously. What are we standardising? Standards and best practices, guidelines, recommendations, and what's what. There's a whole bunch of standards out there now. There's ISO 20387, which has now been approved as they can be moved in around the world and forming the basis of our accreditation programs. Prior to that, though, there was a whole bunch of work, including work coming out of the OECD, our International Society for Biological and Environmental Repositories, looking at best practices, and lots of best practice documents were coming out. NCI, National Cancer Institute, had best practice documents coming out, all of which form the basis for now what we're doing in the ISO 20387. ISO 20387 is part of a standards group TC 276, and within it there's a whole bunch of other little frameworks being set up. This is probably an old slide, by the way. It's 2018, so it's moved on since then. Lots of different standards been brought up, all sort of under this banner of biobanking and biotechnology has been brought in. And so there's lots of different frameworks there. I'm not a part of that. I do know that ISO 20387 has most recently been through a review in Glasgow, and I'm just literally receiving reports now on what came out of that and the changes that may take place and the developments. It's a 4-year review. The 5 categories of biobanking accreditation that we look at when we do an assessment: very basically, is about worrying about the collection and acquisition, pre-analytical testing and preparation, the storage facilities, of course, value-added techniques such as DNA preparations, growing cell lines, making them into organoids, all those sorts of things, and obviously, ultimately, the distribution. It's about samples going from one person to another person. The biobank facilitates that. They all look, this is what we're looking at, it's a dynamic process, not just a sample stuffed in a fridge, all right? And we also see biobanks as infrastructure. And I think we're really at that point with biobanking, that it's not just seen as a research project or something that researchers do when they want to get a cohort together or a collection together, or just to put a few samples away just for a rainy day when they don't know what to do with them. There's actually core infrastructure that has to be part and found within our health and our university environments. Moving forward, recognition of biobanking. Why should we go through accreditation? Why does accreditation matter? Very briefly, by having been accredited to a particular organisation, there's quite a number of biobanks out there being accredited these days, and there's a New South Wales here, we have a certification program that many of us have signed up to. It gives us recognition of our biobanking practices. But one of the things I come up with is why. Why do you want to go through that if it doesn't allow you to do something else? If you go get accredited, you want to be able to do something that you couldn't do when you're not accredited. If you get certified, you get a license to drive a car, you're allowed to drive your car. Right? If you don't have a license, you can't drive your car. Right? Why are we using this? And this is one of the questions that a lot of biobankers are actually asking. We have recognition of our infrastructure. These are the sorts of freezers that we have in our place. You have fast and nimble and flexible and accessible systems. And also we talk about the responsibility of shifting purpose to the source. In other words, where biobanking becomes accredited, the people who are actually giving the samples out, pathology departments mostly, surgeries and so on, have to start considering their role in the accreditation process and the biobanking process. They can't just sort of throw you a few samples and not take any responsibility for what's going on. And we've worked out in our system, and something that I'm a bit passionate about, is actually better for us to push than to pull. If we talk about pull-out biobanking, this is where there's a whole bunch of samples stuffed in a fridge somewhere, and a researcher has to come and somehow, through a whole series of regulations and applications, extract those samples, give up their firstborn so they can get the samples they need to do their research study, right, through an ethics process. Whereas push-out biobanking is hospital-driven or source-driven, where it's actually, they have a mechanism and infrastructure in place that pushes those samples out to facilitate research. We see that with biobanking. Pull-out biobanking is researcher-driven. Samples move from the source to the bank somehow. Project-centred, cohort-focused. It's an additional process. It requires clearances. It has detailed consent, and it's built on goodwill and hope and unknowns. Whereas push-out biobanking is hospital-driven. The source is the biobank. All right? It's patient-centred, it's focused project, it's a process that has add-ons. We're the ethical gatekeepers of those samples. The general consent, it's built on standards, it's routine. And you can see that with pull-out biobanking, for that to work, it requires regulation. Whereas with push-out biobanking, it requires facilitation. And if you have an accredited biobank within a hospital, you'll facilitate research. One that has trust and knowledge, and everyone knows what it's there to do. You can then facilitate research. Moving on, because I'm running out of time, reproducibility, as was mentioned before. The samples are not being reproducible. We're wasting a lot of money with having samples rotting in fridges. And there's lots of different papers out there talking about these 6 factors affecting reproducibility, the mechanism of systems for sharing raw published data, et cetera, et cetera, et cetera. Data repositories and biorepositories need to be made robust so that they are sharing and not an impediment to reproducibility. And finally, just another area of interest of mine is the evolution of biobanking. We've heard about Industry 4.0. Well, biobanking's gone through that too. There's Biobanking 1.0, which is focused on how many samples can you stuff in a fridge. Biobanking 2.0 is about standards. How do we actually get some quality standards in what we're doing? Biobanking 3.0, sustainability. How do you actually start getting business models within your biobanking so that you can actually sustain the activity, and what does that mean? And more recently, we've gone to Biobanking 4.0, with biobanks characterised by systems. Focus on how biobanks interact with the data generation process, the sequencing and the high-end analyses that go on, the deep molecular characterisation, the image analysis, all the stuff that we heard about into this morning's AI talks. All right? And one person once said, why do we give all our samples away? Well, there's a very good reason why you give your samples away, because you're not the only one with the questions. Everybody has questions. They all have data. So let's open them up, get the data out, and allow everyone to explore so we learn new things. So Biobanking 4.0 is a new strategic way of thinking. Biobanking is as an informatics problem. The reason why we want the samples, they provide information. It's not a pathology problem anymore. It's becoming an informatics problem. It's marked by the arrival of AI and machine learning, and it's a dynamic process that less reliance on storage, but more reliance on having a process to generate the data, datify things, and use that data, have it all linked together, and ultimately have it linked back to the patient donor so they have some agency in what's going on in the process. It's a distributed system, and it's determined by the connections and transactions that we can take place, and enables us to personalise our response, our research response and our interrogation to the actual individual and the donor. 7 fundamental lessons. It's not about the verb. It's all about the verb. It's not about the noun. All right? Improved healthcare is utterly dependent on research. Biobanking is founded on relationships being formed between, and trust, those things that we don't need, that we don't talk about enough. Biobanks are facilities, hospitals, not fridges. All right? Biospecimens are packages of information. Biobanking is about distribution, not storage. And success with accreditation must consider all these things and bring these things together. So with that, I think I've made my time. I'll hand over to Helen.
So Biobanking Victoria is actually located southeast of Melbourne, and we're part of Monash University, but physically located at the back of Monash Health campus. This location is ideal for us because it gives us the opportunity to engage with academic researchers from the university, but also the clinicians at the hospital who require biobanking services. So even though we have gained accreditation in 2021, we've got a long history of biobanking, where we actually started in 1993 biobanking. And from that period, now we host over 52 individual biobanks, and we store over 5 million samples in our biobank. In addition to collecting and processing and storing samples, we've also distributed hundreds of thousands of samples worldwide to research projects, including international collaborations. So I wanted to use an example here of where we've used our resource and explain to you how important biobanking is. In this instance, we've used an existing resource. So biobankers understand the importance of having well-annotated and high-quality biobank samples available for research. And in this example, which we've actually conducted in our own lab, there was a project called BRASSTRAP, and the aim of BRASSTRAP was to refine the risk assessment and penetrance of new mutations associated with breast cancer that are now commonly used and in diagnostic settings for women. So what we've done here is we gathered 30,000 DNA samples that had been collected over a 20-year period in family cancer centres and in our own resources. And we brought these from women who had been previously diagnosed as BRCA1 and 2 negative, but had a family history of breast cancer. We put them over a new gene panel and then passed the results through our bioinformaticians and returned results. So for clinically actionable results, they went back to the family cancer clinics, and those results were returned back to the women. For the other results, they've gone back through to international experts, reviewed, and then now the results have been uploaded into EviQ clinic guidelines and are used as part of the new guidelines and best practices for the management of these women. And this was only possible with samples that were over 20 years old, but well annotated and high quality. With the uptake of precision medicine, biobanking services that fall under a quality management system is becoming ever more important. As Dan has explained, while Biobanking Victoria was set up to be able to facilitate the movement of samples that are collected in various settings like clinical trials, inner health services, or even from PI-driven research. And we aim to transition those samples from the place of recruitment right through to cutting-edge research. So, based off this need, we seeked accreditation. And our main drivers was really to promote well-designed study resources, to uplift all our biobank services for our federated facility, because remember we've got 52 biobanks that we're hosting, to reduce the need for ad hoc collections from clinic settings, which is what Dan was talking about, the freezer at the end of the corridor. We also wanted to increase the engagement between biobanks and industry and pharma, and give them the opportunity to be able to work directly together. So part of our journey to accreditation the first part was really to perform a gap analysis of our existing operations against the 4 pillars of the standard. And this was used to identify what documents needed to be written and what needed to be edited. At this point, it actually gave me the opportunity to sit back and review our existing practices and looked at all the literature that was out there. And that included quite a range of literature, including guidelines that Dan talked about, published commercial documents, methods, validated methods, and even additional other ISO standards. And with the aim to uplift what we were currently doing, so we're working at best practices, and also to be harmonised with what other methods that were used for other large biobanks around the world, so that we can all share our samples on a global scale. It also gave us the opportunity to introduce a well-accepted SPREC code, which is a way of it's a nomenclature code that you can apply to your samples, and that also will facilitate the sharing of our samples universally. So what did this look like? It ended up being that we had to write over 300 control documents, and we structured it in a 4-tier system. And in general, we had procedures that that talked about what we do, and then we have protocols to describe how we do it. Having written all these documents, our next step to accreditation was to demonstrate technical competence, as we've heard in a number of talks today. So one of the things that we needed to demonstrate was that we used qualified equipment and consumables when we're in our biobanking service, and that we are using validated protocols that are fit for the purpose, that are reproducible, robust, and produce homogeneous and stable products. We also need to demonstrate that our personnel were competent, and to do that, we undergo regular training and assessments, including participating in internal and external proficiency testing. And this is to ensure that our processes and our products are intra and inter-reproducible. And one of the main other factors is also to qualify our products. Are they fit for purpose? So to do this, every product that we produce is tested using a specific, a protocol specific for the project, obviously, or the sample, and they're tested at the time of production, but also whilst they're in storage, because we want to make sure, or we want to ensure that whatever we're distributing still remains fit for purpose. Remember, a lot of these samples are stored for 20, 30 and 40 years. So are they fit for purpose even when we distribute it? So once we achieved accreditation, it doesn't end there. We have to go have continual conformity assessments, and we do this through internal audits, either by ourselves, which is we're the first party, and we do this quarterly. Every 3 months, we'll have one sort of kind of assessment, be it on a process or equipment or so forth. We also invite a secondary party who's independent of our labs to come in and also be part of our internal assessments, and then we also have our regular assessments through NATA. One of the things that's slightly different to a lot of other biobanks is our operational model. For Biobanking Victoria, we are not responsible for the recruitment of participants or the collection of material. We actually take responsibility of the samples once we receive it, process it, storage it, and to do distribution. That makes us a little bit different to a lot of other biobanks. So the institute who holds the ethics governs the samples and also is responsible for access to the samples. So people who want the samples that we store do not come directly to us, they would go to the institute. As Dan said, there's a lot of dynamic interaction between all this. So we worked with the institute to provide them with the biobanking advice. Since we've been doing this for a long time, we develop methods together. We often sit on the access committees, and there's a lot of to and fro's. So from the time that we have got accreditation, we have now started up 11 biobanks under Biobanking Victoria. We're supporting 6 clinical trials, and we've got 5 currently onboarding. So what are the benefits of accreditation? Really, for me, the main benefit of accreditation is having confidence in our operations, and we achieve this by using validated standardised practices, by having competent staff, and having reproducible processes and products that we know are fit for purpose through our assessments. We also fulfil requirements sought by a lot of our collaborators, and these requirements is often driven by funding bodies, governance, industry and service providers, where they're specifically asking for their products to be coming from an accredited laboratory. In addition to this, is one of our main reasons why we were seeking accreditation, was to be able to increase the engagement between biobanks and commercial and pharmaceutical industries who require products coming from an accredited laboratory. And in the biobanking world, this is very critical. We have been able to attract new biobanks, which helps us in our sustainability. Thank you.
Just really quickly, thank you very much, Dan and Helen. I'm just going to talk a little bit about how NATA actually became involved in the industry. In 2021, biobanking was recognised by ILAC, such that as an activity for which accreditation bodies such as NATA could grant accreditation, and it was to ISO 20387. It follows very familiar standard format for other standards, but it does have the acquisition, storage and distribution key aspects of biobanking in it. Just in terms of biobanks that are around the world, there are 33 biobanks accredited by different accreditation bodies. There are 16 in China and 8 in the USA, which I thought was a very interesting stat to bring up. In 2023, as part of our APAC evaluation, NATA became the second member of ILAC to have biobanking recognised as part of its MRA. It was only timing that prevented us becoming the first one, but yes, so we're very proud of that. We're active members of the Biobanking Working Group and the current review of the standard, which was going to include organoids and AI. We're going to be participating in the upcoming evaluator training, so that for any other people, any other accreditation bodies around the world who actually want to have biobanking added to their MRA, we'll be able to actually help with that. And it's actually recently also been added to the MOU that we have with the Commonwealth Government, which means that as part of, we become the experts that they go to as part of safeguarding Australia's national interests. So a big thank you to Dan and Helen, and I will leave it there.
What do you do once you've done all your testing or distributed the specimens? How long do you keep them for? When do you sort of get rid of them? How do you get rid of them? Because there could potentially be biohazard as well. How does that work after the event?
I'll work backwards. Biohazard, we treat every sample as having potential for that, so we treat, and as in standard pathology type care, you'd be wearing gloves and PPE and things like that. Depends on what you're going to do with them. If you're using things like sonicators and things like that to mince samples up, you'd be wearing masks and things like that. So it's all the usual sort of stuff. You treat them as potentially having some sort of infection, but not, high-end sort of, you don't have to wear the full moon suit type thing. The next question, how long do you keep them for? You keep them for as long as they're going to be able to allow you to answer questions. And as Helen's sort of example is a really great one, 20 years, sample sitting in there. 20 years means that not only do you have the sample, right, treatments have moved on, but by that stage you've also got the full quota of clinical data for that patient, those patients, and you can see the full outcomes. So it's a really rich, really valuable set of data there. So you want to keep them. We're dealing with samples, paraffin-embedded tissue, that goes back to the 40s. And we're talking about rare paediatric cancers, and if you want to make what we call a tissue microarray, and you have collation of these things, you're only seeing 2 or 3 of these patients a year. It takes a long time to build up a little cohort, but if you can go back further, and we've gone back into the 40s and 50s, and therefore you can start actually making some really sizable collections of rare tissue that is really valuable when you're trying to understand a disease which specialist hospitals around the world are trying to work with, and don't really. And it's getting into the high end, the depth of data, like the sequencing and the image analysis and all the stuff that get the people with AI and machine learning really excited. You need lots of data, so you need lots of samples. So the art of the biobanker is to make sure that they can get as much out of these samples as possible to answer those questions. There will come a time where a sample will not be useful because it's been sitting around or it's not been treated properly, which is one other reason why we need standards and accreditation to preserve them as long as possible. And we always have, and we have a bit of a, I don't know whether a hoarder mentality, but we do have that sort of thing that we might learn something new about this, it's like, I might be able to use that one day, sort of mentality sometimes.
In turn, you build more laboratories?
No, we actually engage more with researchers and get more samples out the door.
You would build more of the facilities as opposed to one in Australia.
There's big biobanks around the world, some
Yeah.
very big ones. I think Dan had a picture up. I mean, and that model that's there that holds the millions of samples, there's a couple of those. But it's the data that's associated with it that you could actually see ultimately when you talk about digital biobanks or data biobanks. You just share the data. You don't actually need the sample. So there's some...
Sorry.
Sorry.
Do you also deal with overseas biobanks and their data also? So it's not just Australia-wide, it's worldwide to see if there's any...
That's one reason why the standards are...
Yeah. That's one of the reasons we were looking at being harmonised with what's happening elsewhere. But also, I mentioned that we're part of a lot of international collaborations or consortia where we're pulling data from many different countries, particularly for rare diseases, to be able to answer research questions. So we're all pulling our resources from all around the world to answer one specific question.
Okay, we might call that a close there. Thank you again, our panel, Daniel, Helen and Gillian.